Here we describe the modulation of the lymphocyte potassium channel Kv1.3 by a membrane-penetrating, joint-targeting immunomodulatory plant defensin. Please focus on the graphical abstract on the bottom right. We started with two plant defensin, first called EGK1 from oil palm, and the second called VVK1 from grapevine. We synthesized EGK1 and VVK1, these 47 amino acid peptides, and determined their structure by X-ray crystallography. And then we generated an analog of both these peptides called EGK5, which is remarkably stable. When EGK5 is applied to mammalian cells, it rapidly enters the plasma membrane, where it binds to the lymphocyte potassium channel Kv1.3. It also depletes in the membrane the phospholipid PIP2 and thereby decreases the number of Kv1.3 channels available for gating. And this causes the current to run down, shown in the diagram. Kv1.3 plays a critical role in T cells, especially in terminally differentiated T cells called effector memory T cells, or TEM. And by EGK5 shutting down this Kv1.3 channel, it suppresses the proliferation of these TEM cells. Using a labeled version of this peptide, we did PET-CT studies in rodents and found that EGK5 has a strong propensity to enter small and large joints. Because of this joint targeting behavior, we tested it in an animal model of rheumatoid arthritis. After the onset of disease, we started the animals on EGK5 given once every 2 days. And as you can see on the bottom right, the drug was extremely effective in shutting down arthritis. Toxicology studies done showed that the peptide was safe at 10 to 100 times the therapeutic dose. The alignment on the top left shows that the plant defensins share sequence similarity to toxins from scorpion venom that are known to block Kv1.3. The plant defensins are held tightly by 4 disulfide bonds. 3 of these bonds are shared with the scorpion toxins. The 4th bond links the N and C termini of the plant defensins. On the top right, you see the crystal structures of VVK1 and EGK1. They are remarkably similar. And at the bottom right, you see that VVK1 and EGK1 are structurally very similar to scorpion toxin, calliotoxin in this case, which blocks Kv1.3 potently. We then generated an analog of VVK1 and EGK1 called EGK5, based on molecular modeling and molecular dynamic simulation. EGK5 is remarkably stable, as shown on the bottom left. It is resistant to the proteases trypsin, chymotrypsin, pancretin, as well as pepsin not shown. It's stable at pH 2 to 10, and it's stable in human and rat plasma. Scorpion toxins block Kv1.3 by binding to a shallow basin at the outer mouth to the channel pore. Since EGK5 is sequenced and structurally similar to the scorpion toxins, we tested it on Kv1.3 expressed in mammalian cells, and we used the patch clamp. If you look at the currents on the top left, EGK5 at 1 micromolar has no effect on Kv1.3, whereas the exemplar scorpion toxin, in this case margotoxin, completely shuts off Kv1.3 current at 1 nanomolar. Competition studies between EGK5 and margotoxin show that the plant defensin did not bind in the outer shallow basin, as shown by the dose-response curve of margotoxin, which is the same in the presence or an absence of an excess of EGK5. So despite its sequence and structural similarity to the scorpion toxins, EGK5 and the other two plant defensins, BVK1 and EGK1, do not block KV13. We next tested the ability of EGK5 to bind to purified human KV13, and we found indeed that it bound in a specific manner. And then we used bioluminescent interferometry, not shown here, to determine the affinity of binding to be 500 nanomolar. So it binds but does not block. Plant defensins have a propensity to enter plasma membranes of mammalian cells, and it uses a motif involving 3 arginines. The diagram shown in the bottom left shows EGK5 with the 3 arginines shown, arginine 38, 39, and 40. To test membrane entry, we made a labeled version, EGK5 TAMRA, and tested its ability to enter mammalian cells expressing KV13 using confocal microscopy. And as you can see, the peptide enters the membrane within about 3 hours. It doesn't enter the cytoplasm to any great extent. It stays in the membrane, and it does not cause cytotoxicity because of this membrane entry, either in the fibroblasts expressing KV13 or in activated human T cells. Since EGK5 binds KV13 and enters plasma membranes in about 3 hours, we examined the effect of longer exposures to EGK5 on KV13 currents. If you look at the currents on the top left, EGK5 treated cells, the KV13 current amplitude reduces with time. This decay is not seen in cells incubated with control medium. This time-dependent decay, this rundown, is quantified in the next graph, where in red you see that KV13 currents decay over time in EGK5 treated cells, whereas in blue in control cells there is no such decay. This type of rundown for other channels is due to depletion in the membrane of the phospholipid PIP2. PIP2 plays a critical role coupling voltage activation to channel opening. So we tested the effect of PIP2 on EGK5-induced rundown. Cells were exposed to either medium or EGK5 for a prolonged period, and then they were patch clamped in the presence or absence of PIP2 containing liposomes. In control cells, there is no KV13 rundown, and PIP2 has no effect. In EGK5 treated cells, KV13 runs down with time, and PIP2 prevents this rundown. So in summary, EGK5 enters the plasma membrane. It binds to KV13. It depletes PIP2 locally, and thereby reduces the number of KV13 channels available for gating, and then this causes the current rundown. This effect is specific because EGK5 doesn't affect other channels. 4 are shown below, KV15 and KV11.1 in the heart, KCA 1.1 in muscle and in joints, and KCA 3.1 in T cells. Not shown, we also tested it on a large number of receptors, transporters, enzymes, and other channels, and EGK5's effect is selective for KV13. KV13 plays a critical role in T lymphocytes, especially terminally differentiated T lymphocytes called effector memory T cells or TEM cells. So these cells are implicated in autoimmune disease. So we examine the effect of EGK5 on antigen-driven proliferation of TEM cells, where we measured proliferation by thymidine incorporation. As you can see, when you add the antigen, there's a dramatic increase in proliferation, and EGK5 causes a suppression of this proliferation. We measured the pharmacokinetic and biodistribution properties of EGK5. Shown on the top are PET-CT images of fluorine-18 labeled EGK5, and they show that the peptide targets small and large joints. Other cysteine-rich peptides have been shown to exhibit the same type of arthrotropism. And shown on the bottom are two such peptides, CDP09R, which is a joint-targeting peptide, versus CDP29, which does not target joints. And the difference is the positively charged electrostatic potential of CDP09R. Now, if you look at our plant defensins, EGK5, which targets joint, and EGK1 have a very strong positively charged electrostatic potential, whereas VVK1, the grapevine peptide, is much less positive and therefore should be less effective in going to the joint. Since EGK5 targets joints, and also because it is immunomodulatory through its action on effector memory T cells, which are implicated in autoimmune disease, we tested EGK5 in a rat model of rheumatoid arthritis. We induced collagen-induced arthritis in rats, and after disease onset, we started treatment with either vehicle or EGK5 given by subcutaneous injection every alternate day. And as you can see from the graph on the top left, EGK5 treatment dramatically reduced the clinical score of disease. The pictures on the right show that it reduced bone erosion determined by X-rays, and the histology section show that EGK5 treatment reduced inflammatory infiltrate into joints, reduced cartilage damage, and reduced bone destruction. Interestingly, VVK1, which is less likely to enter joints, was less effective in treating arthritis in this model. Toxicological studies were done showing that the peptide was safe up to 10 times the dose when given repeatedly over a 2-week period. In summary, EGK5 is a cysteine-rich, membrane-penetrating, joint-targeting, remarkably stable peptide analog of plant defensins from grapevine and oil palm. The peptide modulates voltage-gated Kv1.3 potassium channels and T lymphocytes by a distinctive mechanism. It enters plasma membranes, binds to Kv1.3 and depletes PI2, and in this way reduces the pool of Kv1.3 channels available for gating and thereby causes Kv1.3 rundown. Through this channel modulation, EGK5 suppresses antigen-driven activation of effector memory T cells that are implicated in diverse autoimmune diseases. EGK5 accumulates in small and large joints. This arthrotropism is related to its positively charged electrostatic potential. Consequently, EGK5 effectively treats disease in a rat model of rheumatoid arthritis. EGK5 is an attractive candidate to be both a therapeutic because of its channel modulatory and immunomodulatory properties, as well as a carrier to deliver anti-rheumatic drugs such as steroids to joints for the treatment and pain relief of arthritic disease.